So it is my pleasure to introduce Dr. Martha Bulyk. Dr. Bulyk received dual undergrad degrees from MIT, both in mathematics and biology. She received a PhD in biophysics from Harvard University, where she works with Dr. Church Group. Shortly thereafter, she began as an assistant professor in Harvard, and she's currently a professor in the Division of Genetics in the Department of Medicine. And is also a professor in pathology at Brigham Women's Hospital of Harvard Medical School. Dr. Bulyk has been named an inaugural Louis and Elizabeth Mass Endowed Chair in the Genomic Medicine at Brigham Women's Hospital. She's also co-chair of the Harvard Biophysics program and is an associate member of the Broad Institute of MIT. As well as an associate member of Dana-Farber Institute for Cancer. She has had many multiple recognitions for her work. I'm just going to name a few of them. In 2005, Dr. Bulyk was named one of the 35 MIT Technology Review's annual competition to select the top 35 innovators under 35. She was elected 2023 AAAS Fellow by the American Association of Advanced Science for distinguished contributions to the understanding of transcription using computational and molecular techniques for the identification of cis-actin elements. And finally, I will end up by saying that her group focuses on studying transcription factors, DNA regulatory elements, regulation networks, and that beyond her research excellence, Dr. Bulyk has been recognized by the 2025 Brigham and Women's Hospital Education Award, highlighting the broad positive impact that she has had on her trainees and the broader communities. So without further ado, please, Martha, get us started.
Wonderful. Thank you very much, Mark and Inés, for that lovely introduction. And it's my great pleasure to speak in today's webinar. So today I'm going to focus on the interactions between transcription factors and their DNA binding sites, which are a primary step in regulation of gene expression. To identify such interactions computationally, one typically analyzes genomic or more recently synthetic sequence for matches to a transcription factor DNA binding site motif. And ultimately what we'd like to have are predictive models that are mechanistically interpretable, meaning which TFs bind where, under what conditions, and how. We'd like those models to be accurate enough so that we can predict the effects of non-coding variants, such as depicted here, or transcription factor coding variants, which I won't talk about today. A non-coding variant is typically scored for its effect on a transcription factor binding motif. But then we're left challenged in interpreting variants that fall outside of known motifs. Furthermore, various investigators have commented on the apparent discrepancy between in vitro and in vivo transcription factor DNA binding data. However, depending on who you talk to, the opinion is either that the gap is large or actually not that large at all, and that it likely depends on the specific transcription factors or conditions, data models, and analysis methods. Today, I'll tell you about how we're going beyond transcription factor binding site motifs, that there are additional cis-regulatory encodings in the genome that specify transcription factor binding, and I posit that improved in vitro data on transcription factor binding combined with improved computational models will narrow the apparent gap, regardless of how large or small it may be, between in vitro and in vivo transcription factor binding data. In a study we published a few months ago in Nature, led by my postdoc Shubham Ketan, we developed a new high-throughput technology for assaying transcription factor DNA interactions. And the title of the paper gives away what we found, but I'll get to that. Our new technology, called PADIT-seq, is an in vitro solution-based reporter assay where the output is directly proportional to a transcription factor's DNA binding activity. Briefly, a constitutive T7 promoter drives expression of a transcription factor DNA binding domain fused to an alpha tag. Following in vitro transcription and translation, the DNA binding domain then binds to a candidate DNA binding site and recruits T7 RNA polymerase via an anti-alpha antibody, which increases downstream reporter gene expression. Here we assayed binding against a library of all possible 10 base pair binding sites, or all 10 mers. Barcodes in the reporter gene identify the binding sites that were bound. As a negative control, we assayed a no DNA binding domain control. And different DBDs of interest were assayed in separate wells in 96-well plate format. To benchmark PADIT-seq, we assayed 4 human transcription factors and 2 yeast factors, HOXD13, NKX25, TBX5, EGR1, and FO4 and CBF1. For the sake of time, I'll highlight just a few of our results for a few of these TFs. We found that PADIT-seq data are highly concordant with mitomi-derived KD values with detection down to 0.1 micromolar for EGR1. Shown in red are those sequences that we identified as being significantly bound by PADIT-seq as compared to the no DVD negative control, and we call those significantly bound binding sites active at an FDR of 5%. Comparing the PADIT-seq data to universal protein binding microarray enrichment or E-score data for 8-mers, shown here for HOXD13, we see that PADIT-seq is much more sensitive in detecting lower affinity sites that are harder to distinguish from nonspecific sites in, say, PBM data. We also compared the PADIT-seq data to HT-SELEX data and found that PADIT-seq detects hundreds of lower affinity sites that are missed by HT-SELEX. Both high and low affinity transcription factor DNA binding sites play important roles in gene regulation. A number of papers have been published over more than 20 years from both my group and other groups describing various important roles of low affinity transcription factor DNA binding sites in gene regulation, in particular their sensitivity to transcription factor levels for precise gene expression patterns during development. In our recent study, we found that ChIP-seq peaks, depicted here for one, are enriched for having multiple overlapping active k-mers, depicted here. Shown here, for example, at this HOXD13 ChIP-seq peak, there are 6 consecutive immediately overlapping active HOXD13 8-mers. This phenomenon, where multiple transcription factor binding sites overlap each other, is distinct from homotypic clustering, in which multiple non-overlapping binding sites for TF are located within a cis-regulatory element. Across all HOXD13 ChIP-seq peaks, we found that the ChIP-seq peaks were significantly enriched for having a larger number of overlapping active k-mers as compared to random length-matched genomic regions. Overall, around 87% of HOXD13 ChIP-seq peaks contained at least 6 overlapping active 8-mers. Across all these HOXD13 ChIP-seq peaks with at least 6 overlapping active 8-mers, a high-affinity 8-mer tended to be the central TF binding site and was flanked by nucleotides that created overlapping lower-affinity HOXD13 binding sites. To further investigate whether the consecutive 8-mers may be functionally important, we inspected the evolutionary conservation of the corresponding genomic regions. We found that the 13 base pair genomic regions corresponding to 6 consecutive HOXD13 8-mers were significantly more conserved than the flanking genomic regions, and we obtained similar results for ChIP-seq or ChIP-nexus peaks for the other TFs that we profiled in the study. For further validation of our results, we analyzed ChIP-nexus footprint data on TF genomic occupancy that were available for 2 of the TFs that we assayed, CBF1 and FO4, and we saw longer footprints that increased in length in accord with increasing numbers of overlapping binding sites. We next examined the effects of non-coding variants and found that non-coding variants alter transcription factor binding by perturbing multiple overlapping binding sites. Shown here are 2 examples of SNPs. Here are the SNPs, shown in light blue here in the DNA sequence, don't appear to overlap the known DNA binding motif for the affected TFs, HOXD13 and EGR1. And towards improved interpretation of non-coding variants, we found that PADIT-seq data outperformed other data types and models in predicting the effects of non-coding variants on TF binding. Overall, we saw that the SNPs that altered a larger number of overlapping DNA binding sites had greater effects on binding signal according to SNP-SELEX data. So the traditional model of how a TF finds its target DNA binding sites involves a combination of 3D hops and 1D sliding, and our results suggest that the intrinsic property of high affinity sites to have overlapping lower affinity sites confers them with a previously unrecognized mechanism by which they essentially are providing an energetic sink for transcription factor binding. So in addition to assays of TF binding to naked DNA, my lab has also been examining TF binding to nucleosomes. Transcription factors that can bind their recognition sites on nucleosomes are often referred to as pioneer factors. Their binding then leads to the opening of chromatin and enables other co-regulatory factors to bind their nearby target sites. No surprise, many pioneers have been shown to be master regulators of various cell types and to control cell fates. A few years ago, UC Typela's group surveyed over 200 TF DNA binding domains for binding to nucleosomes in vitro, and those nucleosomes were built using completely randomized DNA sequence. Based on their data, they classified TFs according to their positional preference of binding to nucleosomes, with most factors exhibiting N binding. However, it's been seen that pioneers bind only a subset of their motif occurrences within putative regulatory regions in the genome. Therefore, we asked, do nucleosomal sequence context features regulate TF binding to nucleosomes? The study was led by my senior lab member, Luca Mariani, and was published last year in Nature Structural and Molecular Biology. Here we developed a technology that we call PIONEAR-seq. Briefly, we tile across one or more nucleosomal positioning sequences with a random 20-mer tile moving along every 3 base pairs. Here we tiled across 3 different genomic nucleosomal sequences and also across the synthetic WIDM 601 sequence, which has high affinity for the histone octamer and is often used as a model in studies of TF nucleosome binding. Our total library size was around 4 × 10 to the 13th unique DNA sequences. We then pooled all those sequences together and assembled them with histone octamers to create mononucleosomes. We then used the nucleosomes to perform in vitro selection for binding by a particular TF of interest, expressed as a full-length fusion to GST. After each round of selection, we again assembled nucleosomes, and after completing 4 rounds of selection, we sequenced the bound DNAs from each round and also the starting input library. Using the PIONEAR-seq approach, in the study, we assayed 10 proposed pioneer factors. From the resulting sequencing data, we then calculated the k-mer enrichment as the normalized k-mer counts for the GST-TF fusion protein divided by the normalized k-mer counts for GST-negative control. And here the enriched k-mers correspond to the DNA sequences that are bound by the transcription factor. We then examined the positional preference of pioneer binding within nucleosomes. Shown here as an example are our data for binding by CBPB to its main DNA binding site sequence within the randomized 20-mers that we tiled across the WIDM 601 nucleosome, shown here in the lower row. These heat maps depict the relative preference of CBPB binding across the length of the nucleosome libraries, stretched out from the beginning of our first random 20-mer tile to the last one. The darker the color, the more preferentially the TF bound at that position. Shown in the lower row, indicated by the blue box, is preferential binding, as we can see here, towards the right end of the WIDM 601-based nucleosomes. And shown in the upper row as a negative control is the heat map for CBPB binding to naked DNA, where one doesn't expect to see much positional preference. We then compare those data against the PIONEAR-seq data generated using the 3 different genomic sequence templates. And there we found that where within the nucleosome a transcription factor binds, rather than being a simple intrinsic feature of a TF, instead depends on the nucleosomal sequence context. Interestingly, we found that the same end of the genomic sequence nucleosomes was bound preferentially by multiple different transcription factors, not just CEBPB. And the consequence of these results is that the preferentially bound end of the nucleosomes depends on the nucleosomal sequence and not just on the TF. We then asked what might control the end binding preferences of TFs to nucleosomes. We noticed an earlier study by TJ Haas lab in which they found that sequence-dependent DNA local flexibility controls asymmetric unwrapping of the nucleosomes. We therefore asked whether DNA flexibility might also drive sequence-dependent end binding preferences of pioneers, allowing TFs to bind to the transiently unwrapped DNA. To investigate the sequence feature, Katrina Liu, a student in my lab, evaluated the bendability of the nucleosomal DNA based on the predicted sequence cyclicalizability score over 50 base pair windows, tiling across an input sequence. And we see that overall, WIDM 601 is much more flexible than the 3 genomic sequences that we assayed, and that the different sequence contexts also show different degrees of flexibility or rigidity across their lengths. And these results are overall consistent with our hypothesis that the TF binds to the more rigid ends of nucleosomes, as shown here, for example, for the CX3 and NRCAM nucleosomes. And for verification of these results on an independent experimental data set, Katrina then analyzed the Typla Labs and HT-SELEX data for TF binding to nucleosomes built on completely randomized DNA sequence, from which we classified all of the transcription factors in their data set into ones that were either end binding versus non-end binding. And we found that the end binding TFs were significantly enriched for binding preferentially to nucleosomes whose DNA is more rigid towards the ends as compared to the more internal portions of the nucleosomes. To see if the DNA flexibility also modulates CE-BPB binding to nucleosomes in vivo, we then analyzed CE-BPB binding sites within ChIP-seq peaks that overlapped with nucleosomes according to MNA-seq data in K562 cells. Looking across all 10,000 such ChIP-seq peaks, shown here in red, as compared to nucleosomes that have the motif but that were not bound by CE-BPB, shown in blue, we see that there is indeed the same trend for CE-BPB to bind sites within the rigid ends of nucleosomes. So in summary, in this study, we found that the nucleosomal sequence context can alter the positional preferences of PIONEER binding and that DNA bendability controls TF binding to nucleosomes. So today I told you about two recent studies from my group, each of which revealed another layer of cis-regulatory encoding in the genome going beyond TF-DNA binding site motifs. In the first study, we found that multiple overlapping binding sites determined transcription factor occupancy. And in the second study, we found that going beyond immediate motif flanking sequences, nucleosome-level DNA bendability controls TF binding to nucleosomes. With these new tools in hand, we're now working to develop additional new technologies to learn what additional chromatin features may help to explain and predict transcription factor DNA binding in vivo. Finally, I want to thank current and former lab members, as well as our collaborators in Phil Cole's lab at Brigham and Women's Hospital and Harvard Medical School. I already highlighted the key people behind the studies I told you about today, and we're very grateful for our funding from the NIH, which supported these studies.
Martha, I love everything you presented. I will go back to how you started your talk, which is the goal is to find a predictive model that is also interpretable. And I know this is highly complex, but I was really interested in all your findings, particularly the DNA bendability, and have you guys tried? So first off, is there a way to find a model that can predict that? And just from combining sequence plus maybe nucleosome positioning, and then this yes or no, but basically, can you incorporate this and find like a better outcome for predictive models of transcription and TF binding?
Great questions, Inés. So we used a tool developed by TJ Haas lab that was trained on loop-seq data. So loop-seq data were trained on 100 base pair sequences for their ability to circularize. And so they developed a deep learning model that scores for experimental reasons, nature of the data, scores over 50 base pair windows. And so we used that tool, their model, to score 50 base pair windows, and then we just slide along 50 base pair, every base pair, 50 base pairs, average the scores. And so that's how we get a proxy for bendability or flexibility. So, all right, how do we use that? Does that alone answer everything? No, there's more to it. There's more complexity in the DNA sequence. We're hoping to be able to develop improved models to be able to combine that with various experimental data types to develop something that's going to be predictive for where transcription factors are binding in vivo. Not there yet.